Well, thank you. It's such a treat to be able to talk to a responsive audience like all of you. And before I start, I want to say that I wish that I could tell you that the work that I'm going to talk about was initiated by insightful, informed hypothesis testing. But as you will hear, in fact, it was initiated by an accident in the laboratory, and I'll get to that in a few minutes. It's, I think, a good story, and I hope you do too. So, as you probably know, the immune system evolved to generate protection for all of us against infectious pathogens. But in fact, in this class of diseases, of which multiple sclerosis is one of them, the immune system is actually causing the problem. There is an aberrant immune response to self-tissues. And there are over 80 different autoimmune diseases that have been described, and all of them have differences in terms of the target tissue that's attacked and in some of the underlying immune mechanisms that mediate the disease. But one of the things that is very clear in many of the diseases, not all of them, is that there is a sex bias. And in fact, most of these autoimmune diseases in which there's a sex bias, there is a predominance of females developing the disease. And in this slide, I have a graph of some of the more profound examples, including lupus, which shows a 90% susceptibility or predominance in women, Graves' disease, and rheumatoid arthritis, which also show strong female bias, as well as multiple sclerosis. Type 1 diabetes, although there are mouse models in which there are sex biases, does not show a sex bias at all. There have been a multitude of studies looking at the factors that influence this female predominance. And some of these include studies of hormone influence, and in fact, it's a well-known observation that women who are pregnant and have MS very often exhibit a remission of their symptoms during the third trimester of pregnancy when the pregnancy hormones are highest, but they tend to show a rapid relapse after delivery. There are also studies showing that prolactin has a protective effect, but these studies showing that female hormones have a protective effect are actually counteracted by a whole variety of studies showing that they have pathologic effects as well. And as I will talk about later, testosterone appears to be consistently protective in a variety of both human and mouse studies. Environment also plays a role. It's well known that epidemiologic studies have established that growing up in a climate where there is limited sunlight can predispose one to development of MS in susceptible populations, and this is thought to be due to exposure to vitamin D. There are also studies showing that the microbiota of males and females is quite different, and that this can alter the immune responsiveness of males and females and either confer protection or susceptibility. What I am going to talk about today is a signaling pathway that's called c-KIT. This is a receptor that's on most hematopoietic cells, and actually when it's triggered by its ligand stem cell factor, c-KIT drives the development of many immune cell lineages, and I will talk to you about how we think that c-KIT is influencing sex-dependent disease in females and protection in males. All of these factors come together to alter the genetic landscape of cells, and in fact, it's thought that they drive sex-dependent gene expression patterns in males and females. So, for those of you who don't know, multiple sclerosis is an inflammatory disease of the central nervous system, and it is the most common demyelinating disease in the world. There are over 400,000 individuals that are diagnosed in the US and 2.5 million worldwide. It's commonly diagnosed at an early age. Often it strikes young women in their childbearing years, between 20 and 40. And it gives rise to a whole variety of neurologic symptoms. This is a disease that, as I said, is an autoimmune disease, and T cells are essential for initiating the inflammatory damage. And what this figure shows is a cartoon of a healthy nerve on the top, and it shows the nerve cell body, and it shows nerve axons that extend from that nerve cell body into the tissues, and these are the structures that actually conduct nerve pulses into the tissues. And these nerve axons are covered by a myelin sheath, which is a protein made by a cell that's resident in the central nervous system called the oligodendrocyte. These proteins wrap around the nerve axon and help and are, in fact, essential for normal saltatory nerve impulse conduction. In multiple sclerosis, T cells and other cells actually target the myelin. They cause disruptions in the myelin sheath and sometimes actually cause transaxonal damage, and this leads to the variety of neurologic deficits that occur in this disease. There are several FDA-approved therapies for this disease, but all of them have severe side effects or complications, and there is really no therapy that can cure multiple sclerosis at the present time. So multiple sclerosis is a variable disease. There are several common forms. I'm going to talk about two of them. The first is the most common form that affects about 90% of individuals diagnosed with this disease, and it's called the relapsing remitting form. It is most prevalent in women, and it attacks young women primarily, and is characterized by episodes of neurologic symptoms interspersed by episodes of the remittance of those symptoms. And over time, the symptoms get worse, and oftentimes the periods of remission get shorter, and this can evolve into a more progressive disease. The other form of multiple sclerosis is another form is primary progressive multiple sclerosis. This is more often diagnosed in men, and it's diagnosed at a later age in men. And so the point of this slide is to tell you that in multiple sclerosis, not only is there a sex bias in incidence, which is estimated to be 3 to 4 times more frequent in females than males, but there is also a very profound difference in the clinical presentation in males and females. So I'm an immunologist, and I do immune assays. And as a teacher of immunology, I know immunology can be sometimes unnecessarily complicated. So I thought that I would use a few slides to give you a refresher on some of the components of the immune system that are relevant to my story. So this is the first one. All of you probably know that immune cells can be categorized into 2 major classes: the adaptive immune cells, which are made up of T cells and B cells. And these cells have specific receptors for antigen and are really, when they're triggered, expand into large populations. They're long-lived, and they form the basis for immunological memory that's utilized in vaccination, production, et cetera. There are also populations of resident cells that mostly reside in tissues that are called the innate immune cells. There's a whole variety of cell types that each have specialized functions in the immune system. And they are actually the first defense against invading pathogen. One of the concepts that I need you to take home with you, at least, or keep with you during my talk, is that these populations are not really distinct, and that in fact there's a lot of crosstalk between innate immune cells and adaptive immune cells, and innate immune cells are absolutely required for the function of adaptive immune cells. Some of these cells serve as antigen presentation cells to T cells and allow the T cell expansion I talked about. Others of these cells produce various cytokines that promote the expansion, the function, and the localization of T cells into specific tissues. Today, what I am going to talk about, in addition to T cells that are initiating the autoimmune response in multiple sclerosis, are two populations of cells, mast cells and innate lymphoid cells, that are members of the innate immune cell community. And what I am going to tell you is that these innate immune cells are cells that we think have a very critical role in determining susceptibility to autoimmune demyelinating disease. And the caveat is I'm using a mouse model, but I do want to say that almost all of the drugs that are in use right now have been derived from studies in mouse models, so that's my defense here. Okay. So the second concept that I want to convey to you is that T cell responses are quite diverse. And many of you who have taken immunology have learned this concept that a T cell that has never encountered antigen before will encounter that antigen on an antigen-presenting cell in a lymph node or spleen where the T cell is primed. And when this T cell is primed, it undergoes expansion and differentiation, and in fact, the differentiation pathways are diverse. I'm showing 4 of them, but there are several others. These are the major ones. So T cells can differentiate into functionally distinct cells that are called, unfortunately, T helper 1, T helper 2, T regulatory, and Th17 cells. And these cells are defined by the hallmark cytokines that they produce. So Th1 cells produce gamma interferon. Th17 cells are defined by their production of IL-17 and GM-CSF. Th2 cells are defined by the production of IL-4 and IL-10. And very amazingly, it was found when these cells were discovered that they have very important and distinct roles in protective immunity. So Th1 cells in a perfect immunological universe are well equipped to deal with intracellular pathogens such as viruses. They help cytotoxic T cells kill virally infected T cells by virtue of their gamma interferon production. In contrast, Th2 cells are well equipped for dealing with extracellular pathogens, particularly worm infections. T regulatory cells actually have a major role in downregulating an immune response, so they're important in keeping that immune balance. And Th17 cells are also important for extracellular pathogens, particularly some bacterial infections. But one of the things that became clear is that these distinct differentiation phenotypes of T helper cells also have pathogenic roles. And in fact, Th1 cells are associated with autoimmunity, as are Th17 cells. And Th2 cells, if they're uncontrolled, are major players in allergic responses. Now, to connect to what I said in the last slide, the polarizing cytokines that come from innate immune cells drive the different differentiation fates of these T helper cells. So IL-12, if it's produced in the environment where this T cell is primed, will drive Th1 cells, and TGF-beta and IL-6, for example, will drive the Th17 response. What I'm going to tell you in a few minutes is that cells of the Th17 phenotype and cells of the Th1 phenotype are important in determining the sex-biased immune responses and susceptibility to autoimmune disease, autoimmune demyelinating disease. Okay, and then finally, one of the things that's curious about inflammatory diseases of the central nervous system, including multiple sclerosis, is that for a long time it's been thought that the central nervous system, and by that I mean the brain and spinal cord, are what we call immune-privileged tissues. That is, unlike other peripheral tissues in our body, immune cells don't circulate freely through those tissues. And the idea was that mechanisms to prevent that normal circulation and immunosurveillant role of immune cells were not allowed in the central nervous system because inflammation is so devastating. The tissues, individual cells of the central nervous system are in large part unable to regenerate, and so if you have immune inflammation in the central nervous system, it is more devastating than if you have it in other tissues where you can get easy regeneration, or more easy regeneration of tissues. So one of the major mechanisms that controls this immune privilege, so to speak, is the blood-brain barrier, and probably most of you have heard of this. And really what it is just specialized blood vessels. These blood vessels have very tight junctions. Very tight junctions between the cells that actually make up the blood vessels. They also are covered by different kinds of cells, including cells that extend foot processes along the surface of the outside surface of the blood vessels called astrocyte foot processes. So the consequence of this is that it is very difficult under normal circumstances for immune cells and large molecules to actually access the central nervous system. However, in multiple sclerosis and in the mouse model that we study, pathogenic T cells gain access to the central nervous system, and they initiate this immune-mediated damage to the myelin sheath. And this is a cartoon that actually is adapted from a rendition by Steve Miller, who is one of the major multiple sclerosis researchers here at Northwestern and in the world, I would say. And what this shows is that T cells encounter their antigen in the lymph node or spleen. And in the case of a pathogenic response, these T cells are either Th1 or Th17 type cells. They are prompted to proliferate, and then they move into the circulation. And as they move along the circulation, they enter vasculature that makes up the blood-brain barrier. They encounter cells that are associated with the blood-brain barrier vasculature, including mast cells and other monocytic cells, and they are reactivated. This prompts the expression of a lot of chemokines and cytokines that break down this blood-brain barrier, opening the junctions and allowing immune cells to pass through. The T cells get in. They can re-encounter their antigen by resident or influxing antigen-presenting cells. They produce GM-CSF, which is, again, a hallmark of that Th17 response I told you about earlier. And that GM-CSF recruits a whole variety of immune cells, including monocytes, that then produce reactive oxygen species that actually are the main molecules that carry out the damage to the myelin sheath. And so you get this damage to myelinating nerves, and you get the neurologic symptoms that follow that. So our laboratory has used a model of multiple sclerosis called experimental autoimmune encephalomyelitis. And there are different models that recapitulate different features of the various forms of the human disease. We have been using the SJL mouse model for a number of reasons. The first is that it's a model of relapsing remitting disease, and this is showing a prototypic course of relapsing remitting disease, and as you can see, it's much contracted compared to the human disease. This model is induced by immunization, and we actually use peptides that make up one of the myelin proteins, and this induces a T cell response. And the disease is scored over time on a scale from 1 to 5, where 1 is a limp tail and 4 is hind and forelimb weakness. The other advantage to the SJL mouse model is that it exhibits very profound sex differences. Females are susceptible, and males show much lower incidence. They have less severe disease, and they show a less consistent relapsing pattern. So one of the things that we observed, but other people actually observed this back in the 90s when we couldn't do these assays as well, is that males protection is not due to the lack of a T cell response. In fact, the protection is due to a qualitatively different response in males and females. And now that you've had your immunology primer, you can appreciate that females mount a Th1 and Th17 response when you immunize them with myelin protein, whereas males mount a Th2 response to the myelin protein that in this context is non-pathogenic. And what we are showing here is experiments done to actually analyze T cell responses in males and females by looking at the hallmark cytokines that are produced by those. And you can see here that males, when you immunize them, and we're looking at lymph node cells here, but we also see the same thing in the central nervous system, males produce very good Th2 type responses to myelin proteins, including both IL-4 and IL-10, whereas females produce GM-CSF and IL-17 responses. So this fits with susceptible females and protected males. So the big question is, why do males and females generate different types of immune responses? And as I told you, there's been a lot of work done looking at various factors, X chromosome dosage, hormones, microbiota. And we kind of hit a wall probably back in the late 90s, and there's only been a little information trickling out since that time. And in fact, Abby Rusi, who has done the bulk of this work and is a super student, went to a conference and presented this work, and somebody came up to her, and this was in the early stages, and said, I worked on that for 20 years. He said, it's going to kill your career. Don't continue, don't continue. So it's been a really difficult problem to understand. So what happened with us is that we were using this c-Kit mutant mouse, which we call WWV, and surprisingly, it offered a lot of insight into the underlying mechanisms that caused males to produce a qualitatively different anti-myelin response compared to females. So WWV mice are mice that have a mutation in c-Kit, a receptor for stem cell factor. And this receptor, as I said, is very important in the development of most hematopoietic cells. However, this mutation isn't a total mutation. There is still some signaling that is allowed through this receptor in these mutant mice. And the consequences of this mutation are such that there are two types of cells that don't make it through development untouched. Those are mast cells that I talked about, and type 2 innate lymphoid cells. Other cells, and we've done extensive characterization of this mouse model, appear to be normal. T cell numbers, dendritic cells, macrophages, all the other innate immune cells, B cells. So really the major defects in immune cell functions are mast cells and type 2 innate lymphoid cells. So when we started studying the effects of c-Kit on disease, back in 2000 actually, a graduate student at Emory University did this project, what we noticed, and we noticed it over and over and over again, was that wild-type females developed a relapsing remitting course of disease, but females with a mutation in c-Kit had a much milder disease, and we started to study all of the mechanisms that contributed to this pathogenic effect on disease by c-Kit positive cells. However, I moved to Northwestern and had a new student. Oh, and let me just say that we never used male mice because male mice don't get sick. And if male mice don't get sick, why use them? Because we can't study what makes them sick, and we were so focused on what makes females sick that we never really thought to go back, and other people had done that, compared males and females. So I had a student, she was a brand new student. She didn't know that male mice didn't get sick, but more importantly, When mice are small, it's very difficult to distinguish their genitalia. And I have a hard time. I have missexed animals, and so it was not unsurprising, or not surprising. So this student came in and pulled a whole cohort of animals and did her experiments. And when she did that, we just got patterns all over the place. And we had gone 9 years getting this particular pattern of disease in wild-type and c-kit mice. So when we finally figured out that the mice, that the reason for this crazy pattern of disease that didn't make sense was that, in fact, she had pulled cohorts of male mice. We looked at that pattern, and in fact, we saw exactly the opposite pattern that we see in females. So this was a happy accident, and this set us on the course of trying to understand why this mutation has completely different effects in males and females. And you can see here that wild-type males get a mild form of disease, whereas c-kit mutant mice get a more severe clinical course, show a more severe clinical course. The funny story associated with this was it was picked up by NPR, who was soliciting nominations for an award called the Golden Mole Award for Accidental Brilliance. And I got an email from a guy at NPR, and I thought it was a joke. And I went online to find out what the Golden Mole Award for Accidental Brilliance was. And in fact, and I apologize for those who are not, who are politically sensitive, but women, we've been the target of these kind of comments all our lives, right? So, in fact, the golden mole is a rare animal that lives in the deserts of Africa and in some places in Australia. And it's blind, and it burrows under the sand, and really very rarely comes out into the daylight. And scientists that were studying this mole had been noticing that it had this very shiny iridescent coat, like Donald Trump's hair. And they could not understand what the reason was that led to the evolution of this very shiny coat, because it couldn't attract mates because these animals were blind, and it wasn't really camouflage because they lived down in the sand and never came up, so they didn't have to be worried about predators. And it turned out, as I studied it more, this same features that give this coat its shiny color also provide this very smooth ability for them to smoothly move through the sand and catch their prey that live in the sand. So it's the accidentally brilliant acquisition of this coat. So that's where the award got its name. And we were not the winner, but we were a finalist. And if anybody wants to look this up, it's funny. We were number 6, a case of mistaken genitalia. So I do contend that really a lot of good discoveries come from accidents. And if you stop and look at, wow, why is this happening, you can learn a lot. And in fact, my student, when I first told her, I said, you didn't immunize some of them, and she pursued it. So to her credit, it led us along an interesting path. Okay, so one of the first things that we did was look at the immune responses of c-kit mutant WWV mice and compare them to wild-type mice, males. And just like susceptible females, mutant males generated a Th1 and Th17 dominated response, in contrast to the non-pathogenic response in wild-type males. When we thought about these c-kit mutant mice, as I told you, there were two types of cells that were deficient. One was mast cells, and the second was this type of cell that was not really described until the late 2009, called type 2 innate lymphoid cells. And these cells were very interesting to us because they are c-KIT innate immune cells that express cytokines, including IL-4, IL-5, IL-9, and IL-13. They reside in immune border sites, like skin, lungs, and guts. But most importantly, they had been described in some detail as critical players in eliciting Th2 type responses. So it piqued our interest. So these cells, again, have been described in allergy models, never in an autoimmune disease. They are defined by a number of cell surface markers, including the IL-33 receptor called ST2 and c-KIT. And when they're activated, they produce cytokines that promote a Th2 response. And these cytokines, particularly IL-13, actually act on Th2, developing naive T cells to drive them to become Th2 producing cells. So when we went back to the wild type and mutant males, we asked what happened to these cells in the context of immunized mice. And in fact, what we saw was that in both the lymph nodes and the central nervous system tissues, brain and spinal cord, when you immunize these animals, in wild type males, you got this very marked activation profile that was not present in the mutant mice. And again, this is true in both the lymph node and the central nervous system tissues. And of course, you would expect that because these cells are deficient in mutant mice, they wouldn't be activated. But we were quite surprised to see that these were activated in the local draining lymph nodes as well as the central nervous system. So we went and asked, are ILC2s functional in females? And if not, is that why they default to a Th17 pathogenic response? So when we compared ILC2 activation in females and males, and wild-type females and wild-type males, as well as mutant males, we saw that, indeed, there was not this activation of these ILC2s. And by activation, I'm defining that as an mobilization into the lymph node and central nervous system, as well as IL-13 cytokine production. So there's a much reduced mobilization into the relevant tissues, and the fewer cells that are present make a much reduced IL-13 response. So these cells don't appear to be normally functioning as they are in males. So again, the next question is, are there sex-determined differences in ILC development in females or in function? So Abby Rusi did a very extensive characterization of ILC2s in a whole variety of tissues, and I'm just showing you a few examples here. In the bone marrow, the lymph node, and the brain, where they reside at some low frequency in unimmunized animals, there is absolutely no difference in the steady state populations between males and females. This led us to the question, are there sex-determined differences in the ability of these cells to respond to ILC2 activating factors? So the cells are there normally in both males and females, but they're not functioning in immunized mice. So what Abby did was set up an experiment where she treated animals with IL-33, a ILC2 activating factor, and what this does is elicit an expansion of these cells in the bone marrow. She then isolated the bone marrow, and she purified these cells by flow cytometry analysis. And then she treated these with a variety of ILC2 activating factors. And after 6 days, she looked to see what the consequence was for function and expansion. And in fact, again, there were absolutely no differences in the ability to expand these cells. Here's the male ILC2 profile by flow cytometry analysis, and in the female cells. And we looked at proliferation, and they look absolutely identical. She also looked at cytokine responses, and as you remember, in immunized mice, females don't make a good cytokine response, but if you give them the factors that they need, they act just like males. So this told us that although the populations are intact in female mice, they don't mobilize to the lymph nodes and central nervous system, and they don't show a strong production of Th-promoting cytokines after immunization. However, if given those activating signals, such as IL-33, ILC2s from males and females respond similarly. So we concluded that there's just no intrinsic defect in the female-derived ILC2s, and that wasn't the reason that they were not activated in immunized mice. So the next question was, do males and females have differences in their production of ILC2 activating factors? And the paradigm for how ILC2s are activated, again, was developed in the allergy field. And it was developed in models where mice were given protease allergens, and it was thought that damage to the lung and gut epithelium by these protease allergens actually elicited a variety of ILC2 activating factors here that I've showed here. So IL-25, IL-7, TSLP, and IL-33. And these factors act on the ILC2s to promote their proliferation, their mobilization to tissue, and their cytokine production. We first focused on IL-33 because it is a factor that has been clearly shown to induce Th2 responses, and it is the most selective of all the ILC2 promoting factors. And again, ILC2s are defined by high expression of the IL-33 receptor. And when we looked in mice that were immunized with myelin peptides, so both males and females, we looked at day 6 and 14 in various tissues, including the lymph node, brain, and spinal cord, for the expression of IL-33, and we did these experiments by real-time PCR. We saw that males had much higher expression of IL-33 in all of the tissues examined compared to females. And one of the things that I'm not going to talk about at great length here, but is quite surprising and striking, is that if you look at the array of genes made by females, there's a different pattern. Females just don't lack IL-33 expression. They have increased expression of other sets of genes, including pro-inflammatory genes that have been highly implicated in multiple sclerosis, like TNF and IL-1 beta, whereas males make very little of those, express very little of those genes. So next question was, what cell types are producing these producing this IL-33 in the context of disease? And we know that epithelial cells make it, but it had been reported by Paul Bryce, who's at this institution, as well as others, that mast cells express IL-33. And we've also shown that dendritic cells and macrophages can produce IL-33 under activation conditions. So we asked, do mast cells, in the context of disease, produce this cytokine? And indeed, they do. We showed this in a couple ways. The first was to use mice that are deficient in mast cells and measure the IL-33 response. So here is unimmunized mice, male mice, and mice at day 6 post-immunization, and we're looking at whole lymph node production of IL-33. If the mice do not have mast cells, they do not express this at any appreciable amount, and females have much reduced IL-33 expression, as we saw before. We also looked at mast cells that we isolated from the meninges, which are membranes that cover the brain and spinal cord and are very mast cell rich tissues. And you can see, I think, yes, that male, wild-type male mice have a nice population of mast cells as defined by c-KIT and Fc epsilon receptor expression, whereas the mast cell deficient ones do not. And these mice we looked at the IL-33 expression by these mast cells at day 6 post-immunization, and we see a nice response. So IL-33 protein is being made in the context of disease by mast cells in male mice. We also have a way of deriving bone marrow mast cells, and this is by isolating bone marrow and culturing them for up to 8 weeks in cytokines that promote the differentiation to a pure bone marrow-derived mast cell population. And when we activate these cells, either with IgE receptor or through the toll-like receptor for 5 hours, and look at IL-33 expression, as well as other mediators that we assessed, we can see again there is a male predominance of IL-33, TSLP, and TGF-beta, whereas an enzyme that induces a mast cell protease is indistinguishable in males and females. And along the same lines as the tissue analysis, where we saw very distinct profiles in males and females, mast cells derived from females produce high levels of TNF and IL-1 beta, whereas males have an imperceptible production of that. So obviously the $64,000 question is, why do male mast cells produce more IL-33 than females? And we next tested the possibility that testosterone contributed to these differences. As I alluded to earlier, testosterone is very clearly implicated in protection in mouse models. And there have been studies showing that the onset of MS in males corresponds to reduced bioavailable testosterone that occurs in men with age. And in fact, there have been some very limited studies showing that testosterone treatment of male MS patients actually improved cognitive function and reduced lesion size. The studies in mice are much more compelling, and I've just mentioned a couple of them here. If you castrate protected SJL mice, they become completely susceptible to disease, and their disease course is indistinguishable from females. And if you treat young females with testosterone, you can induce protection. So testosterone was a good candidate for mediating this difference in immune responsiveness between males and females. So we looked at mast cells first and asked if they could express testosterone receptors, and in fact they do. These are mast cells isolated from the peritoneum. Mast cells are tissue resident cells, and they're hard to isolate from tissues, but the peritoneum actually serves as a nice source of mast cells, and also bone marrow mast cells. In peritoneal mast cells, the expression is higher in male-derived mast cells versus female-derived mast cells, but it's fairly indistinguishable in bone marrow mast cells. ILCs do not express the androgen receptor, so we do not think that they're a direct target of testosterone. And this is showing that in this panel, and here is a panel with control cells, including the testes, which has high expression of androgen receptors. So we have a lot of work to do, but as a first step, we have done some experiments, and when I say that we, I mean that Abby and others in the laboratory, to ask if testosterone can directly induce IL-33, and indeed it can. Abby derived male and female bone marrow mast cells, and then she treated these mast cells with testosterone in the presence and absence of an androgen receptor antagonist, and then did real-time PCR to look for the expression of IL-33 at the gene level. And in fact, you see a dose-dependent increase in IL-33, and this is inhibited by flutamide, this androgen receptor antagonist. So what we think is happening is that testosterone is acting on mast cells and probably other innate immune cells that express the testosterone receptor. And we think it has both acute effects in directly driving IL-33 production, but also may change the potential for those cells to produce certain sets of mediators, including IL-33, at the epigenetic level. This IL-33 drives ILC2s in males, and you get a Th2 response that predominates to myelin, and the mice are resistant, whereas this lack of testosterone allows a completely different gene program to be expressed by innate immune cells, which then promotes this Th17 pathogenic response. So the final question we had for this study was, can ILC2s be augmented for therapy? And again, Abby Russi did these experiments where she took female mice that are susceptible, and she induced disease and then treated the animals prior to onset of disease, as well as in the early stages post-immunization, and looked at disease scores. And so in this graph, you see untreated mice, but you see a diminution of clinical scores with increasing concentrations of IL-33. And strikingly, the ILC2 response, which is relatively low in wild-type female mice that are untreated, shows a large increase in treated mice, and this corresponds with a very stunning Th2 response in these mice. Now, that's all fine and dandy if you can identify people who are going to get MS, and if you use some variation of this for treatment, but in fact, the most important test of such a treatment is to ask if it improves established disease, and in fact, these are pilot experiments, but what Abby did was wait until the mice developed discernible clinical disease, and then she treated the mice for 3 days. And in fact, these mice that were untreated went on to begin to relapse, whereas the mice that were treated did not relapse in the same way. And this failure to relapse corresponds with increases in ILC2s and the expression of Th2-type cytokines. So in a situation where you have an established Th17 pathogenic response, you can shift that response, prevent relapses by virtue of generating this Th2 predominant response. So in conclusion, there is differential activation of these innate lymphoid cells in males and females in immunized mice. This activation is driven in part by distinct sex-determined mast cell responses. We do think that other innate immune cells are also differentially expressing this, and we have some data to support that. The protective response in males may be the result of early imprinting by testosterone during development that alters the epigenetic landscape of these cells and promotes IL-33 expression, as well as direct effects of acute testosterone exposure on IL-33 production. And treatment with IL-33 mobilizes ILC2s and switches the pathogenic Th2 response to a protective one in susceptible female mice, which is quite remarkable, I think. So some implications of this work, obviously, it's really important to understand the downstream pathways that are implicated in testosterone protection, because it's not, we're not able to give long-term testosterone treatment to females, especially since those females are generally the diagnosed females are in their prime childbearing years, and even long-term effects of testosterone in males is probably not good. So knowing the cellular and molecular pathways that induce this protection via testosterone is really important. IL-33 is associated with both immune modulating and neuroprotective effects, which is a really important point. And so it may be not only useful in stopping the inflammatory damage, but may have a role in actually regenerating those delicate neuronal tissues. There may be a rationale for exploiting these pathways to treat other CNS inflammatory diseases that have some parallel mechanisms involved. The other important point is that males and females respond differently to some of the currently available therapies. And we found a pathway that's protective in males, and albeit it's mice, it can confer protection in females. Obviously, a big, important next push will be to verify that these pathways exist in humans, and I think we can imagine that it might be possible to identify distinct epigenetic landscapes in males and females and actually use that as a marker of disease susceptibility or resistance in the future. So I thank you for your attention, and I'll take any questions.